Greetings all. This is going to be just a quick video about the different variants that you keep seeing in the news and kind of making sense about what those variants mean and just the kind of the taxonomy and naming of these viruses. Virology taxonomy is kind of a pain in the neck, and there's any number of fairly arbitrary ways to name a virus, and so this hopefully will kind of explain what the variants are. But first, a little refresher. Your coronavirus, in this case, use the virus of the day, binds to surface receptors on your cell, and in this case it binds to the ACE, I should have looked up what it was, angiotensin converting enzyme. It's an enzyme that degrades angiotensin and actually lessens the immune response or the inflammatory response. So it's a negative feedback enzyme, kind of. But anyway, this virus hijacks that system and binds to this enzyme, and that allows it to penetrate the cell. Your cells are lined with these surface receptors, and these are proteins, right? So your cells have these little signature of proteins on their surface. And in this particular case, the ACE protein, I guess it's really the same with all proteins. If you recall the levels of protein structure, and why are we doing this? Well, this is going to factor into how we name viruses and how we classify mutations. Levels of virus structure: you have the primary structure, the secondary structure, tertiary structure, which is kind of the three-dimensional folded shape of amino acids, and then the quaternary structure, which is found in complex proteins. So these are multi-subunit proteins. And see if I can get my highlighter on the right one. So if you go to the primary structure of amino acids, you'll find just the primary structure of protein. You'll find the sequence of amino acids, any of the 20 common protein-making amino acids, and different amino acids, like building blocks you can link together, have different chemical properties, and that enables them to fold into different characteristic shapes. Your DNA genetic code determines the order of amino acids that you link together for any one protein. So you could number these, and if you numbered these, if you unraveled any of your proteins and started with the first amino acid, you would call that number 1, 2, 3, 4, 5, and 6. So cysteine, in this case, I'll have to look at this: cysteine, arginine, alanine, guanine, glycine, glutamate, and phenylalanine. Right, that would be the, if you want to have fun, go to YouTube and look for the longest word, and it's somebody who took the 4.5 hours or whatever it is to name the amino acid structure of the protein Titan. It's just ridiculous. It's, cysteine, alanine, glycyl, glutamate, phenylalanine, but it's 4 hours of that. Anyway, tell you all that to tell you this. So you can name these, number these amino acids. So the coronavirus binds to these surface receptors, these proteins, and this triggers, if you remember, endocytosis in the virus. I'm working with a new way of animating, so we'll see how this works. The virus gets into the cell and replicates inside the cell, filling up the cell, and then ultimately, in this case, lysing the cell and moving on to others. So what changes in the virus potentially can cause it to be more or less lethal, more or less infective, more or less pathogenic, I guess. So higher infectivity and virulence pathogenicity. One thing is how well it binds to the receptor. So it hijacks this ACE receptor, and that triggers the cell to pull it in. And the binding strength or binding affinity for the spike protein on the surface of the virus to the receptor protein, small changes in that amino acid structure or sequence can cause more or less greater or less binding affinity. Another thing that makes a difference is if you have immunity, and so this is the idea of either getting the vaccine or getting the disease, is that you have built up these antibodies in your bloodstream, and so if a virus does kind of seep into your blood or become bloodborne at least, you'll have these neutralizing antibodies that, and these are not to scale, right? These are massive Ys I just printed out. But antibodies will bind to the virus and neutralize it. So other things can contribute to how well a virus infects individuals and how virulent, I guess it is, how damaging it is, but really the ability to bind to the host cell is pretty much the requirement for whether a virus can infect a host, right? That's what gives viruses their specificity. The ability to jump from one host to another or one species to another can be a simple mutation in the surface receptor, or in the spike protein, the virus spike protein, allowing it to recognize a different surface receptor. And then your immune response also, if you have a weak or strong immune response, and if the virus changes or mutates to evade your immune response. So if I go to, and a lot of this is, again, bear with me, this is all new stuff, but ways I'm trying to figure out how to animate this, so you all are the guinea pigs on this. If I go to the kind of a close-up of the virus attaching to the ACE receptor prior to endocytosis, the binding strength of this is going to be important, right? So if you remember back in biochemistry, all the nightmarish pKa values and strength of binding and binding affinity and binding curves and all that awful stuff that you hated, it all becomes actually very important here, right? So how well does the virus actually attach to the host? How strong is that connection? So if I focus just in on this, and I'm going to borrow an image from the web here, you can see the virus, and then down here where that's attached to the blue would be the ACE enzyme, your receptor, and the red would be the virus spike protein, and there's this little R receptor binding domain, RBD. The RBD motif or section on this virus spike protein is going to be found, see if I can use the pointer, this is exciting. I don't know if you can see this or not, but going to be found all throughout here. So this is the receptor binding domain of the spike protein. So the virus establishes this connection with your host enzyme, and this triggers you to intake the virus, or to endocytose the virus. Or if you remember back to that primary sequence, if I were to do this same thing, and I'm just drawing off the line here, but I could draw the amino acid, or write the amino acid structure in this series of linked amino acids, and ultimately get this three-dimensional structure. And if I linearized it, I would have, I should have looked up how big this protein actually is, but I know it's over 500, so well over 500 amino acids long. So what would happen if a virus mutated? During replication, the genetic code changed, and this happens a lot in viruses. Actually, viruses replicate or mutate at the highest frequency known, particularly RNA viruses, which coronaviruses happen to be. So an RNA virus replicates and mutates, changes very quickly. Makes sense because it lives in an environment where the host is constantly trying to kill it. And so a dynamic environment requires a dynamic population. So what if the virus changed, mutated, and a phenylalanine, in this case I just made up one, right, substituted for the alanine on the third amino acid? I would call that mutation if I was to name it. And the whole background to all this is to get to this. I would call that amino acid change an A3F. An alanine used to be there. It was on the third position. It changed to a phenylalanine. Phenylalanine abbreviated F as an amino acid. So I went to Google News and didn't even type in a news search. Just scrolled down until I found the first thing for viruses, and lo and behold, there is a variant with a K417N mutation. 417, not the area code for Southwest Missouri, also 417, but K417N. So what is this? You know, there's a Delta variant. There's all these different names for viruses, right? For the different strains or genealogies, lineages of these viruses. What's the K417N? If you look at your amino acids, your 20 different amino acids, apparently my picture cut off some of these, but thankfully not the ones we're using. K is the code for lysine, and so 417 amino acids into the virus spike protein, there was a lysine. And the virus changed, and instead of a lysine, now there is a asparagine. These are both negatively charged amino acids, but they're different in structure, slightly different in structure. And it turns out that the K417N region in this particular area is a place where antibodies tend to bind. And so a small change in that area can lead to your immune antibodies not binding as well, and which obviously is not ideal. And this has actually been borne out experimentally when combined with other mutations as well. On its own, it's not a terrible mutation, but when combined with several others, it leads to a much weaker antibody response. So, last but not least, second to last but not least, I know I've wasted 11 minutes so far of your day. Variants of the virus. So again, virology, I hate viral taxonomy just because you can classify viruses and they're changing constantly. So virologists can have the luxury of sometimes making it up a little bit. I mean, everything's made up anyway, right? All of our systems are made up in our heads and we use them, they work. Anyway, the two main variants we went for was, both of them came from Wuhan late 2019, early 2020. Variant A and variant B were kind of two major subtypes of this virus. Every significant mutation that produces a significant change in the infectivity, in any quality, major quality of the virus, gets its own separate branch and lineage ultimately. And so you can make sub-branches in and off of these. And so you have like A1, A11, A12. Ultimately, there is a new variant, or fairly new, it's not really new, I guess, but the South African variant, also known as, which was B1.1.1.1, so for like 4 ones, just became C1, and that is the South African variant. And so this is the one that was in the news lately, because it contained these 3 mutations. Again, most mutations are harmless, right? Actually, most mutations are what are known as silent. They don't even manifest in a noticeable phenotype difference, but occasionally some may be, well, it's kind of weird. We call them harmful. The virus would call them helpful. So, depending on, how you want to say it, which end of the microscope you're looking through, right? Known detrimental mutations, and there are quite a few of them, but these are just 3 that are found in this combination of the South African variant, and linked down here is the CDC and the proteogenics, where I got some of the information. There's the K417N, where antibodies bind, right? There's the N, I should have looked it up. I don't even, see, I never bothered to memorize the abbreviations for the amino acids. Because why when there's a chart? And now, oh, this is why. N501Y, so I know there's at least 500 amino acids, right? Because there's a substitution at 501 from an N to a Y. Well, it'd be asparagine, I guess, to whatever Y is. Increases the binding affinity to the host, so it makes the virus recognize your receptor better, if you will. And then exacerbated then by an E484K mutation, which K we know is lysine. Well, this is helpful, right? So you put a negative charged lysine instead of whatever E is, and enhances the effect of this prior mutation. So those are some known ones, and this is floating around in the news, right? You'll see, new variant emerging. This, this has higher infectivity, The CDC, and if you go to the CDC page, you can find this, has ways of classifying the different variants as far as the potential threat that they pose. And they're different, there's, medium consequence, low consequence variants. So these are interesting, but they don't seem to matter as far as how we treat the virus or how well it spreads, the mortality rate of the virus, so forth. The variant of high consequence is really, if you're to look at anything from all of this different thing, right, would be to check the CDC's website for a variant of high consequence. This would be a something where preventative measures, medical countermeasures have significantly reduced the effectiveness relative to previously circulating variants. So this is something where all of a sudden medical interventions are not working as well, right? In addition to the possible attributes of a variant of concern, impact of countermeasures, demonstrated failure of diagnosis, maybe you can't detect it, maybe the vaccine doesn't work as well, right? Maybe this did something where now significantly reduction of vaccine effectiveness. Vaccine breakthrough cases or very low induced protection against severe disease, right? Susceptibility, severe clinical diseases, increased hospitalizations, right? So these are things that the WHO and the CDC in coordination also look for. And currently, if you go right now, as of whatever today is, somewhere in the middle of June, there are no SARS-CoV-2 variants that rise to this level. And so you'll see a lot, maybe the main take-home is, besides if you're interested in how they name viruses and where the variants come from, the names of the mutations, the take-home is you'll see a lot in the news about different variants coming in, this strain emerging, and that's newsworthy. It's scientifically relevant. It's definitely worth keeping an eye on, but it's not worth fretting about necessarily. Knock on wood, right? I mean, obviously, but it's not worth being overly distraught about when there are no variants of high consequence that have been identified. So these are things, they emerging this and this and this concern. I will say this, just to close, the longer this pandemic goes on, the longer this spreads, the more likely that you will develop variants of high consequence. We're basically putting a novel pathogen under some pretty intense selection pressure. When populations have this, and you can get into population genetics, but when a population has a significant decrease due to either a high selection or due to some natural occurring event, the two things that the population can do are go extinct or else rebound and be significantly different. I would just as soon this disease not rebound and be significantly different. So on that note, keep an eye just on the CDC page. Look for, talks about variants of high consequence. Don't get too worried about the different strains that are coming up here and there. They're noteworthy. Some of them are slightly more infectious, slightly less responsive to treatment. This is to be expected, right? You would expect this. All in all, I didn't even expect to have the vaccine roll out until about now, right? So from what I've seen historically, this was, we're doing better than we could have. So that's just kind of and I keep seeing these things pop up in the news, which is why kind of what inspired me to make this video. So keep an eye on for variants of high consequence on the CDC website if you're concerned about any of it, and we'll see you in class.